Hello, my name is Will Price, and I will be doing my presentation on the article titled The Role of Glucose 6-Phosphate in Mediating the Effects of Glucokinase Overexpression in Hepatic Glucose Metabolism. So why is glucose 6-phosphate important? For starters, it is the first intermediate of glucose metabolism, starting from glucose in which glucose gets phosphorylated. Glucose 6-phosphate serves as the branchway intermediate to multiple metabolic pathways. These pathways include glycolysis, pentose phosphate pathway, glycogen synthesis, and de novo lipogenesis, as well as hexosamine pathway. One thing important to note about glucose 6-phosphate is that it responds to changes in glucose concentration and glucokinase activity, which will be shown later on. Now, talking about hexokinase 4 is a very important enzyme in the liver and is more commonly known as glucokinase. This enzyme catalyzes the first step of glycolysis, and there are also three isozymes of glucokinase known as hexokinase 1, 2, and 3. Glucokinase also differs in its ability to not be inhibited by glucose 6-phosphate and having a low affinity for glucose. Glucokinase is also predominantly found in the liver. When also talking about glucokinase, it can be noted that it is also regulated by insulin and glucagon, as well as glucose and fructose 1-phosphate precursors, which results from glucokinase binding with proteins. One specific binding protein is known as JKRP, or glucokinase receptor protein. This protein inhibits glucokinase activity and sequesters it in the nucleus at low glucose levels. Now, when talking about fructose 1-phosphate, it can be noted that it decreases glucokinase's affinity to JKRP and causes it to move from the nucleus to the cytoplasm, which you can see where I've taken it from the slides in the PowerPoint from class. Now, dissociation from JKRP can be a result of elevated glucose 6-phosphate content in the cell. There are also small molecule activators of glucokinase that can increase its affinity for glucose. They also do not cause the dissociation of glucokinase from JKRP, but they do impact liver cell glucose metabolism by activating glucokinase and translocating it from the nucleus. This next slide is going to focus on glycogen synthase and phosphorylase. Now, when talking about glycogen synthase and phosphorylase, we need to know that glucose 6-phosphate is a regulator of these two, and that glucose 6-phosphate's effect on glycogen synthase is regulated by multi-site phosphorylation. UDP glucose, as we learned about in class, is the substrate of glycogen synthase, and glucose 6-phosphate increases the enzyme's affinity for it. PP1, or protein phosphatase 1, is a favored conformation of the enzyme, which is better for dephosphorylation. Now, why is this favored? Because the affinity of glycogen synthase for glucose 6-phosphate increases with dephosphorylation. And now we're going to talk about phosphorylase A versus phosphorylase B. Now, glucose 6-phosphate regulates phosphorylase A by allosteric inhibition and promotes its dephosphorylation. But phosphorylase B is catalytically inactive in the liver and is the dephosphorylated form of A. This results in further activation of glycogen synthase. Glucokinase can also cause activation of glycogen synthase by binding to a dual specificity phosphatase and associated with PP1 in a multi-protein complex, which can cause the dephosphorylation of glycogen synthase. Now, the next part of this presentation will try and show you how glucose 6-phosphate contributes to the metabolic effect of glucokinase overexpression in hepatocytes. Now, how does glucokinase affect metabolism, and what metabolic effects do small molecule glucokinase activators have that can lead to future treatment of type 2 diabetes is the type of questions this will answer. To start off, an inhibitor of glucose 6-phosphate hydrolysis was used called S4048 to alter or modulate the cell content of glucose 6-phosphate without glucokinase expression. Metabolic control analysis will also be used. Now, what is metabolic control analysis? Well, it is a way to quantify the control concentrations and the changes in enzyme activity have on the flux into a pathway. This will come into play later on. The effects of S4048. Now, to determine the role of glucose-6-phosphate, 2 different tests were ran, and these tests were done independently of glucokinase expression. The first test used different concentrations of S4048 on glucose-6-phosphate and determined the rate of glycogen synthesis in liver cells that were incubated with 15 millimoles of glucose. And that means that they use the same concentrations of glucose. Now, the second test used the same concentration of S4048, but different concentrations of glucose. Now, what were the results that came from this? Well, glucose-6-phosphate increase was caused by S4048 inhibition of glucose-6-phosphate hydrolysis, and the rate of glycogen synthesis was increased. And glycolysis was barely affected. Now, S4048 also had no effect on glucose metabolism or glucokinase binding. Depletion of phosphorylase due to S4048. Elevated glucose-6-phosphate causes inactivation of phosphorylase. So wouldn't the increase in glucose-6-phosphate caused by S4048 also cause this? Well, they tested the effects of S4048 on the activity of phosphorylase A. Now, what they saw was that there was a depletion of phosphorylase A between the concentrations of 5 and 35 millimoles. Now, what caused this depletion? So, they tested the effect of S4048 at 5 millimoles of glucose in the presence of dihydroxyacetone and in the absence of it. In the presence, it caused glucose-6-phosphate to increase, and the inactivation of phosphorylase confirmed it was elevated by glucose-6-phosphate. Now, this graph to the right actually shows the concentrations from 5 to 35 and how phosphorylase A is depleting instead of inactivating. And that will lead us to our next slide. The metabolic effects of glucokinase overexpression in S4048. Now, after proving the validity of S4048 as a tool to modulate glucose-6-phosphate content, they wanted to compare glucokinase overexpression and S4048's metabolic effects on glycogen synthesis and glycolysis at different concentrations of glucose, and then correlated the changes in flux with glucose phosphorylation and glucose-6-phosphate content. Now, the results found that glucokinase overexpression increased the glucose-6-phosphate content and glycogen synthesis. And what was also found is that glucokinase also caused increased glucose phosphorylation. And the combined data from S4048 and glucokinase overexpression showed the rates of glycolysis correlated stronger with glucose phosphorylation, while the rates of glycogen synthesis correlated stronger with glucose-6-phosphate content. Next, we need to determine the control exerted by glucose-6-phosphate. Now, three things were determined by using metabolic control analysis, as you learned about earlier. One was the sensitivity of glucose-6-phosphate content to glucokinase activity, which was expressed as the concentration control coefficient of glucokinase. Number 2 was the sensitivity of glycogen synthesis to glucose-6-phosphate content, which was expressed as the flux control coefficient of glucose-6-phosphate content. And the third thing that was determined was the sensitivity of glycogen synthesis to glucokinase overexpression, which was expressed as the flux control coefficient of glucokinase. Now, what were the results? Now, the results showed that the concentration control coefficient of glucokinase was independent of glucose concentration, and interestingly enough, both of the flux control coefficients were higher at 5 millimoles of glucose than at 20 millimoles. Now, what caused this? It was the dual effect of glucose 6-phosphate on phosphorylase and glycogen synthesis. So they wanted to test if the high flux coefficients were because of the dual effect of low glucose 6-phosphate and by an effect on glycogen synthase at elevated glucose 6-phosphate. To do this, they used CP91149, which is an inhibitor of phosphorylase A, and they compared the effects of it on phosphorylase inactivation and glycogen synthesis activation with the separate and combined effects of S4048 and glucokinase overexpression. And what was found was that the phosphorylase inhibitor caused CP91149 caused a greater depletion of phosphorylase A than S4048 and glucokinase overexpression. They also found that the activation of glycogen synthase by S4048 and glucokinase overexpression increased due to increased glucose 6-phosphate content. And they also found that the activation of glycogen synthase by CP91149 was independent of changes in glucose 6-phosphate content. In summary, the things learned was glucose 6-phosphate is intermediate to multiple pathways. It also responds to changes in glucose concentration and glucokinase activity, and it is and glucose 6-phosphate is catalyzed by glucokinase. Glucokinase is also not inhibited by glucose 6-phosphate and has a low affinity for glucose. And glucose 6-phosphate is a key regulator of glycogen synthase and phosphorylase. And glucokinase can activate glycogen synthesis as well. And we also learned about metabolic control analysis and how glucokinase overexpression affects glucose metabolism in the liver. And we learned that S4048 was used in this study, and we learned how it inhibits glucose 6-phosphate hydrolysis, and we learned about the control exerted by glucose 6-phosphate. Now, my first question is, why might the increase in glucose phosphorylation under short-term conditions increase glycolysis during activation of glucokinase? My second question is, based on what was talked about in this presentation, what would be the benefits of the study, and how does it correlate to glucokinase overexpression in the liver? And that would be all. Thank you.